Hi, this is Mrs. Ortega, and I'm here about to take a look at Chapter 11, which is circulation. So the heart is a muscular pump. So this is going to drive the circulation of blood throughout the body. The blood flow is extremely important as we'll see it goes through a closed circuit of vessels to every tissue in the body, delivering oxygen that has come from the lungs and nutrients that's come from the small and large intestine and picking up waste for disposal. And so in this chapter, we'll take a look at some big ideas of the circulatory systems, the human circulatory system, the cardiovascular system, and heart. We'll also look at structure and function of blood vessels and structure and function of blood. So first off, let's take a look at some circulatory systems from some animals. So they have going to facilitate exchange with all body tissues. The simplest of cavities is the gastrovascular cavity that we've seen in other chapters, and this aids in digestion and in transport. And I'm sure you are familiar with, hopefully by now, and tired of that word, but we will continue on and look at other circulatory systems of the open variety and the closed type. And so in open circulatory systems, the heart is going to pump fluid through open-ended vessels. These vessels will be surrounding, will be nearby to bathing tissue cells directly. And so they're going to be around and near organs and bathe those organs. And that fluid will return to a vessel that will then be pumped or lead to the heart to be pumped again. For a closed circulatory system, the heart's going to pump the blood that travel through the arteries. These arteries will go to capillaries, then from capillaries will go to veins and return back to the heart. And so this never leaves a blood vessel. The heart's going to pump blood through blood vessels. These blood vessels we'll learn are arteries, capillaries, and veins. And so we'll see how these two systems are much, much different. And so looking at first at the open circulatory system, an example here is a grasshopper, which you can see has a tubular heart and pores all along, and tubular hearts along the body from head to tail region. And so looking at this open circulatory system, the heart's going to pump fluid through the open-ended vessel. That vessel is going to lead to a region, a cavity that will be open in nature, have fluid, tissues, cells, organs that fluid will bathe directly. This fluid actually has a name, and I will write it for you. It's called hemolymph. We define it differently with a different word to illustrate how much different it is than blood. It's going to have a different composition. It's called hemolymph. And so the blood will return back to the heart through pores. These pores are going to be very structured, very structured for a reason, and that they're going to have these valves that will close. They close right before contraction. That way, when the heart contracts and pumps the blood out, it's going to pump it efficiently. And then after the contraction's over, the valves will open during the relaxation phase to allow for that fluid to fill up the heart. And so lots of organisms with open circulatory systems do depend on body movement to help that hemolymph circulate through their system around organs and things that are not, when it's not circulating back to the heart. So now we'll take a look at closed circulatory systems. These circulatory systems will have a heart with two chambers. These two chambers are going to be defined as an atrium and a ventricle. So atrium is going to receive the blood from the vein, and so this is going to be a temporary storage site for the blood. Then the ventricle is going to take that blood and pump it to the gills for the fish in this example, going out through large arteries. And so we'll learn here that arteries will direct blood away, whereas veins return blood to the heart. And so from arteries, it'll go to arterioles, and then from arterioles, it'll go to the structure called capillaries or capillary beds. And so here's a diagram of a closed circulatory system of a fish that you can see starting with the heart, the atrium, and the ventricle on the ventral side of the fish, the bottom side of the fish here, is going to pump blood, oxygen poor blood to the gills. These gills will then exchange that carbon dioxide for oxygen, and then from there it's going to go to the second round of capillary beds to the body and exchange the materials with the body, and then that will return back to the heart. So what are the key differences between an open circulatory system and a closed one? The open circulatory system is not going to have vessels connecting the heart to the rest of the body. The closed circulatory system will. And another thing is the fluid itself. The open circulatory system fluid is a different composition. It's made up of different molecules, and so that's called hemolymph, whereas a closed circulatory system is going to have blood. And so now we're going to take a look at the circulatory patterns of different animals. So the first one being that fish we just saw. It has a two-chambered heart, so one atrium and one ventricle. This is going to pump blood in a single circulation or a singular pattern. And so there are other ones we'll see that have a double circulation and different circuits called a pulmonary and systemic circuits. And so they also have different chambers to their hearts. We'll see that amphibians and many reptiles have three-chambered heart, whereas birds and mammals have a four-chambered heart. And so here is our fish example again, but in different format of a single circulation. So the atrium will temporarily store the blood, the oxygen poor blood. The ventricle will pump it out, and that will go to the gills. On the capillaries of the gills will exchange the oxygen and the carbon dioxide. And then from the gills, it will go through blood vessels to the rest of the body. We'll see in later cases, usually this actually returns back to the heart at this point, but for fish it does not. And it exchanges carbon dioxide and oxygen on the blood capillaries, and then from there the oxygen poor blood goes back to the heart. That's single circulation. So one pump goes through the gills and then the body, whereas double circulation, there's going to be two pumps that occur. And starting with the right atrium, yes is backwards. Yes, everybody knows. It's due to the fact that if you're looking at it from a patient perspective or the organism perspective, the right side would be the left side and the left side would be the right side. So we're not confusing you on purpose. It has been designed this way for a reason. The right atrium is right here. That right atrium is going to fill up with oxygen-poor blood. It's going to pump it into the one ventricle right here that this three-chambered frog or amphibian has. And then that ventricle is going to pump it out through a artery, through an artery that's going to go to the lungs of the frog, or in some cases skin. So remember hopefully from your dissections that skin is also a respiratory surface for amphibians. And so from there it will exchange the gases of oxygen, carbon dioxide. And so because it's both lungs and skin, they call it pulmocutaneous. And so if you were to break that word down, pulmo will mean lung. And then cutin or cutaneous is going to mean skin. So lung and skin circuit, all right, going to the heart. And so that's going to fill up that blood with oxygen. It's going to lead it back to the heart. So through the left atrium, it's going to temporarily store it. And then it's going to pump it through the ventricle again. And so that is then going to go pump it, the ventricle is going to pump it to the rest of the body. And the body is going to exchange the gas and then return the blood back to the right atrium. And so you may be wondering, well, why do they only have one ventricle? Well, I will tell you that this ventricle is going to be actually partially divided right along this purpley region that you see. There's going to be a ridge there. And so about 90% of the blood is going to be divided. And so it's very helpful in terms of when the frog is on land, it can divide where it wants the blood to go. So on land or in the water. So if the frog's in the water at the time, then it's going to divert all of the gas, to the oxygen, carbon dioxide, the blood, all of that to go to the skin. As if it's underwater, it's not going to be able to breathe through the lungs. And so the skin is going to exchange those gases with the aquatic environment. And so it's a very unique function that fits them perfectly. All right, so looking at double circulation of a bird or a mammal with a four-chambered heart is essentially the same process, except there are a few changes. The first one is that the ventricle is divided into two, so there is a left and a right ventricle. And then there's the circuit. They both have a systemic circuit pumping blood to the body, but now in the four-chamber heart of the bird or mammal is going to have a pulmonary circuit that goes just to the lungs. And so you can see here the right ventricle is going to pump blood to the lung capillaries, which then exchanges it, returns it to first the left atrium and then the left ventricle. All right, through our checkpoint question. What is the difference between single circulation of a fish and the double circulation of a land vertebrate? Well, in single circulation, that blood goes from the gill capillaries to the body capillaries before returning to the heart. So that's one circle that makes. In a land vertebrate, that goes back, the blood travels to the lungs and then returns to the heart a second time before it goes to the body. And so it circulates twice before it's completed its cycle. All right, so now we'll take a look at the human cardiovascular system and heart. So we fall into the mammal category. We have a four-chambered heart with double circulation. And so we have a pulmonary circuit going from the heart to the lungs and a systemic circuit, heart going to the rest of the body. And so the blood flow of the double circulatory system in humans is very similar. We do have some new names, some new structures that we'll look at. And so here you can see the written description, and I will look at the picture and describe it to you in picture form. So now we're going to start with your left ventricle. It's going right there. It's going to pump oxygen-rich blood out of the heart. And so as it's going away from the heart, it's going through the aorta. The aorta is going to lead to other arteries that will then carry the blood to the upper and lower regions of the body. So the head, chest, and arms is one region, and then the abdominal region and legs is another. And so from there, they will exchange gases with the body. So it's going to take in the oxygen, put the carbon dioxide in the blood, and then it's going to return back to the heart. So returning back to the heart from the head, chest, and arms will be through the superior vena cava there. And then returning it from the abdominal region and legs will be the inferior vena cava there. And so that's going to then go to your right atrium. The right atrium is going to temporarily store that until it gets to the right ventricle, where then it's going to pump the oxygen-poor blood to the lungs. And so as it's leaving the heart, anything that leaves the heart is going to be an artery. It's going to flow away from the heart. And so that's a pulmonary artery that's going out of, from the ventricle to pulmonary arteries. The oxygen-poor blood is going to go into the lungs where through the capillaries it will exchange gases. And then from there, those capillaries form veins, pulmonary veins that return blood back to the heart, specifically back to the left atrium. It will go to the left ventricle and be pumped all over again. And so it's important to note in this diagram, in following diagrams and previous diagrams, oxygen-rich blood is noted as red, oxygen-poor blood is noted as blue. In reality, oxygen-rich blood is a very bright red, and oxygen-poor blood is a dark red. So the color coordination is not actually correct, but it helps understanding the process, and so that's why it's done. And so keep in mind here two main ideas: that veins will always return blood back to the heart, and arteries will always carry blood away from the heart, away from the heart, and so that's going to carry it to the body, or in some cases to the lungs. And so away, arteries carry blood away from the heart, veins return blood back. So why does the blood in pulmonary veins have more oxygen than the blood in the vena cavae, which are also veins? While the vena cavae are actually returning oxygen-poor blood back to the heart, and pulmonary veins have just filled up with oxygen from the lungs. And so that's why. So pulmonary veins carry blood from the lungs where it has picked up oxygen, but the vena cava have just returned from the body, returning blood to the heart that's been from the body. So that's lacking oxygen. So I have a video here for you. There's a separate video where you will complete an assignment on. It's a little bit more entertaining video, hopefully, but this is a more educational one that I'll go ahead and play for you here.
In mammals, blood pumped by the right side of the heart passes through the lungs and enters the left side of the heart. This is the pulmonary circuit. Blood pumped by the left side of the heart is distributed to the other body organs and returns to the right side of the heart. This is the systemic circuit. This pattern, called double circulation, ensures vigorous blood flow to the organs because the heart pumps the blood a second time after it returns from the lungs.
All right. So now let's take a look at the heart. So the heart's going to contract and relax rhythmically. This will actually do it automatically on its own. But in other cases, it can be influenced by other things like the nervous system and the endocrine system. But we'll take a look here at how that rhythmic functions. And so during the relaxation phase of the heart, this is called diastole. It is a Greek word that means to place apart or expansion. So expanding the region is going to be relaxing. And so during diastole, the chamber of the heart is going to relax and blood will come into that chamber. During systole, this is also Greek, which literally means to contract. During systole, contractions of the atria will push blood into the ventricles, and then stronger contractions of the ventricles will propel blood into the large arteries carrying blood away from the heart. And so you can see the structure here. The atrium are on top, the ventricles on the bottom. They have valves in between them that are called atrioventricular or AV valve for short, which makes sense, between atrium and ventricle, A and V. And so then the semilunar valves will be valves that the blood goes through after the ventricle. And so you can also see where the pulmonary artery is going to carry blood away from the heart to the lungs, and then the aorta carrying oxygen-rich blood away from the heart to the rest of the body up top there. So the heart valves that you see in that image help prevent backflow of blood, which is making the heart more efficient and pumping and spreading that blood throughout the body, throughout the lungs. So the volume of blood that each ventricle pumps per minute is called the cardiac output. And heart rate is the number of heartbeats per minute. For the average person, there is a number of 72 beats per minute, or BPM for short, 72 beats per minute, and the volume of blood that each ventricle pumps per minute is about 70 milliliters. This is just for your factual purposes, not for studying purposes, these numbers here. But let's take a look at how the heart contracts and relaxes. So first stage is the diastole phase, so relaxation phase, where the atria and the ventricles are going to be open or relaxed, and so their valves are open, and this is going to allow for all over filling of blood in the ventricles and the atria, and so of both sides. And so it's important to note here the semilunar valves are closed. This is wanting to pull in and store blood. Then in the next stage here, about 0.1 seconds, the atria contract. And so this contracting will push blood from those storage chambers into the ventricle. And so from there, the ventricles will contract. So the contracting ventricles is going to push blood from the ventricle out through the arteries. And so those semilunar valves are going to be open. And so in that 0.3 seconds, that process takes place, and then after that, following that, the valves will open and will enter diastole phase where it fills up. And so if you're wondering about the rhythmic beat you hear from hearts, the lub-dub sound, the contracting phase here, when those AV valves close, that will be the lub sound. So that's the result of AV valves that are closing in between the atria and the ventricles. Now if I can write here. And so the dub sound, it actually comes from the third step, where the semilunar valves, when those close, that forms a dub sound. So lub-dub, lub-dub, the beat of the heart comes from the systole phases. And so how does the heartbeat maintain itself, you may ask, and that's through a set of area that relays signal. And so this is in part with the nervous system. There's a region called the SA node or sinoatrial node that will generate an electrical impulse in the atria, and this sets the heart rate or the heart contractions. And so this is also known as a pacemaker. So any person that has to have an artificial pacemaker put in will have an artificial SA node. Another node we'll see in this next image here is the AV node, which stands for atrioventricular. And if you were to guess that it would be along that atrioventricular region or by that valve, you would be correct. So this is going to relay signals to the ventricles and trigger ventricular contractions. And so the impulses can be read on an electrocardiogram, or EKG or ECG for short. So this detects electrical impulses in the heart and can provide data about heart health. And so here you can see the process that occurs in the electrical impulses, and that the SA node pacemaker starts the impulse, it sends it throughout the atria, and then it'll hit the AV node. There's a slight delay there in time as it processes the signal and then sends through specialized muscle fibers or bundle branches to the apex of the heart, so the tip of the heart there. And then that signal will then result in contraction of the ventricles, and this is done by Purkinje fibers. These are, I believe, named after a scientist. Purkinje fibers will contract the ventricles. And so in EKG, there is a distinct electrical signal that can be read and interpreted. And so if it deviates in one position or another from the standard, then it would be a concern for the doctor and the doctors of the patient or scientists of the study. And so looking at EKG is very informative on how the heart rhythm is maintaining itself. And so for those that do need an artificial pacemaker put in, here's an example of one. It's an artificial pacemaker implanted in the chest. And so this is going to be wired to the SA node and can stimulate those contractions, which will then keep the heart in contraction and rhythm to maintain the life of the patient. All right, so here a checkpoint question. A slight decrease in blood pH causes the SA node to increase the heart rate. How would this control mechanism benefit a person during strenuous exercise? is slight decrease in blood pH. So this is going to be because strenuous exercise increases the amount of carbon dioxide in the blood. So if you remember from last section, I believe, we looked at how in gas exchange and breathing, carbon dioxide, when added to blood, causes the pH to drop. And so as the heart rate accelerates, that will enhance the delivery of oxygen to body tissues. And so that removes carbon dioxide quicker as well. And so this also speeds up the breathing rate. And so that's how a slight decrease in blood pH causes the SA node to increase the heart rate. Because the presence of carbon dioxide is going to decrease the blood pH, and this change will be detected by the SA node, and it will will increase the heart rate. And so during strenuous exercise, you're needing lots of energy, and so this needs a lot of oxygen. So that would, and expelling the waste of carbon dioxide, and so that would be beneficial for a person during strenuous exercise. All right, in terms of complications of the cardiovascular system, a heart attack is defined as the damage or death of cardiac muscle tissue. This usually results from a blocked coronary artery. That's something we haven't looked at yet, which we'll be able to see on this next image. A coronary artery is going to be an artery carrying oxygen-rich blood to the heart. So it needs its own supply of blood to survive, for its own cardiac muscle tissue that we looked at before. And so those coronary arteries, if those get blocked, that can result in a heart attack. So damage or death of that cardiac muscle tissue can cause a heart attack. And so more than half of the deaths in the United States are caused by cardiovascular disease, which can be disorders of the heart and blood vessels. So this encompasses a wide variety of illnesses. A stroke is defined as the death of brain tissue. And this is due to the lack of oxygen that usually results from a blockage or a rupture of arteries in the head. And so this is also can be caused by the cardiovascular system, and so it can be classified as a cardiovascular disease. And so the blockage or rupture of arteries in the head results in oxygen not getting to the brain, and so that can result in death or damage of brain tissue. And so patients can have a wide variety of symptoms and a wide variety of range in terms of seriousness of both strokes and heart attacks. Recoveries can be made, but it can also be fatal. So it really depends on what type of damage has been done, how serious the damage is, and if the patient can survive while getting treatment by a professional. Some of these happen so very quick that even with a medical personnel, it's just not quick enough to save the patient, unfortunately. So here we see the picture of the blockage of a coronary artery resulting in a heart attack. So as you can see, these coronary arteries branch off of the aorta, and they cover the whole entirety of the heart and are spread out throughout the whole heart. And you can see a blockage that's occurred with dead muscle tissue there due to the fact that they could not get enough oxygen and could not expel carbon dioxide waste. Other complications are blockage of arteries and blood vessels not related to the heart or to the brain. And so these can be impaired gradually by a disease known as atherosclerosis. So during this, fatty deposits called plaques develop on the inner walls of arteries, and that just narrows the passageway of which blood can flow. And so it can eventually lead to a blockage in that artery, and so that can be very serious. And so here you can see an image of the blood vessel. So there's a layer of epithelial tissue, some smooth muscle, and some connective tissue. And when plaques form, and these fatty deposits on the side of the epithelium that you see there, it can lead to a blood clot, which is also shown. This is networks of platelets, fibrin, and even sometimes red blood cells that can get there. And so recently in the news, you have probably seen how COVID-19 has led to patients having unexplained blood clots that can result in death. So this is a very serious process that occurs in patients and people worldwide. And so it is something very important, but not on this slide in particular, not something I ask you to memorize something from this here. All right, so now we're going to take a look at the structure and function of blood vessels. So looking at blood vessels, we'll first look at the three main groups: arteries, veins, and capillaries. Arteries are going to carry blood away from the heart. They're going to have a thicker muscle because they need to maintain that high blood pressure and constrict and reduce that blood flow. And so veins are going to have thinner walls and thinner muscle, but they have other ways to get blood to return back to the heart against the force of gravity in our lower extremities. And that's through one-way valves. The third one here, capillaries, are your tiny, the smallest blood vessels out there. They're going to have very thin walls made up of that simple squamous tissue. And they're going to function with gas exchange of carbon dioxide and oxygen with the cells and the red blood cells and the body cells. And so you can see the structures here of arteries and veins. They first have a layer of epithelium tissue, then smooth muscle. Arteries are going to have more smooth muscle than veins, and then connective tissue that will connect it to other organs, other body parts. And so looking at capillaries, you can see epithelium layer and then an extracellular matrix layer that will connect it to other fluids like interstitial fluid, as well as other cells, other tissues. Also picture here is the valve in the vein that prevents any backflow. So once the blood goes through that valve, it cannot return. And so that helps in getting the blood back to the heart through the veins. Here on this image, you can see the capillary. So the capillary has a simple squamous layer that is, in terms of diameter, just wide enough to fit one red blood cell at a time. And so this is going to allow for a great amount of exchange to occur between the red blood cell and the tissue cells and the interstitial fluid that you can see here. So how does the structure of the capillary relate to its function? The small diameter and thin walls of the capillary will help with exchanging substances between the blood and the interstitial fluid along with the body cells. So next is looking at blood pressure. So blood pressure depends on the cardiac output, so amount of volume per ventricular pump and the resistance of blood vessels. And so it's best to understand in terms of at what point of where in the blood vessel it is. And so we will see in this next slide that process. In terms of velocity down here, and then pressure here, going through the aorta, it's going to be have a very high velocity as that heart has just pumped it and it's ready to go, it's ready to leave and go to the rest of the body. And so the pressure there is going to be very, very high compared to other stages. So in terms of pressure, we have the relaxation phase of the diastolic pressure and then the systolic pressure, which is the contraction phase. And so it alternates, like the heart alternates. And so that's going to have alternating pressure going back and forth with every heartbeat there. And so it's very high in the aorta, the arteries, and arterioles. But as it reaches the capillaries, it's going to slow down. Like rush hour traffic here is going to go very, very slow through the capillaries. And so the velocity goes to nothing. It slowly decreases in pressure as it goes through the capillaries. And so as it enters the venules, which will then lead to veins, and then from veins to the vena cavae, the systolic pressure continues to decrease as it's been a long time since it's been pumped by the heart, and so that pressure is pretty much non-existent, whereas the velocity does return through the veins in the vena cavae, and that's due to the valves along with muscle movement, which we see in this next picture here. The contracting skeletal muscle will actually, surrounding veins will actually help push that blood flow back to the vena cava, which will then go back to the heart. Another connection between heart cardiovascular problems and blood pressure is something called hypertension. So this is a very serious cardiovascular problem that is essentially an elevated blood pressure. And so looking at blood pressure, you measure both systolic and diastolic pressure. And so an elevation in either one of them can lead to hypertension. And so prolonged hypertension is a major cause of heart attack, heart disease, stroke, and kidney failure. And so we'll take a look at how to measure blood pressure and hypertension, the stages of hypertension. So our checkpoint question here says, if a woman's blood pressure is consistently 140/75, does she have hypertension? The answer will be yes. Even though her one of those is normal, her systolic pressure is elevated, and so that leads to hypertension. And so for most people, the normal blood pressure is 120/80. For adults around the age of 20, a normal blood pressure is actually around 120/70, according to newer textbooks. And so how do you measure blood pressure? Well, you can use a machine, or you can go old school and use a inflator cuff and a stethoscope, which you see here. Important side note about the machines, the arm machine is the most accurate. The wrist machine, while accurate, it does depend on your positioning of your wrist, and so that can lead to inaccuracy. And so that is typically not recommended by medical professionals, but it's still a valuable source. So it can still give you a reading, an accurate reading, but it's not always perfect. And so that's why it's recommended to use an arm machine. But anyways, how to do that, you might ask. And that through old school ways is to first inflate, put the cuff on the arm at the same level as the heart. inflate the cuff until you no longer hear any noise, or until above 120 is easy to do. And then at above 120, you start slowly deflating the cuff. And so when you're deflating the cuff, you're going to be listening in through the stethoscope. And so as that pressure decreases in the cuff, it's going to compare to the pressure in the artery. And so when the pressure in the artery equals the pressure in the cuff, at that level, the blood is going to start to flow. And so you'll hear a very faint trickling sound. And so when that trickling sound starts, that's when you know the blood pressures are equal, and that's your first reading. And so typically that would be 120 millimeters of mercury. And so then you you keep deflating that cuff. And so very slowly when deflating that cuff, we do not hear the trickling noises anymore. When the sounds stop, that's when the cuff is in the same state that the artery is in, and that's a relaxation phase. And so that will be hopefully around 70 for adults in that age range, around 20, or 80 for anyone, and the national average is 80. For our textbook purposes, though, we'll use 70, and so 120 over 70 is the goal for blood pressure. And so the normal blood pressure level for systolic is less than 120. So remember, that's your contracting phase. That's going to be a higher pressure, versus diastolic, which is less than 80 or 70 ideally. And so that's a relaxation phase, that the pressure would be lower there. So there are stages of pre-hypertension and stage 1 hypertension and stage 2 hypertension that are shown here. And so looking at that checkpoint question again, if a woman's blood pressure is consistently 140 over 75, does she have hypertension? And she does. Stage 1 hypertension, because her systolic pressure is 145, and the range here is 140 to 159. And so she falls into that category, even though she's less than 180 on her diastolic. Alrighty, so looking at capillaries. Capillaries can be regulated through sphincters. This is very important in terms of functioning, but as far as studying goes, you don't need to understand the precapillary sphincters that you see here. It helps regulate the blood flow through capillaries, and so this can also help in terms of constricting and dilating blood vessels. Regulating the capillaries will also be for that part too. But for capillaries, what we will want to know are that they are the only blood vessels with walls thin enough for substances to cross. And so as we know, oxygen and carbon dioxide freely cross the capillary wall to and from the red blood cell to the interstitial fluid and to body cells. And so any excess fluid that's lost, so fluid and red blood cells can actually be lost through the capillaries, and that gets returned through lymph vessels, through the lymphatic system. And so here you can see a scanning electron micrograph of a capillary in the lungs. And so here, this would be part of the alveoli. This is your capillary, and then these are your red blood cells right here. So it's very neat to see in real life pictures what that looks like. But diagram picture here, looking at the movement through the capillary. There is the blood pressure and osmotic pressure that's involved. Any fluid or cells or platelets that get lost, well, they will enter a lymph vessel and return back to the circulatory system. All right, in terms of, skip this. Last common idea or big idea here is the structure and function of blood. So now let's take a look at blood. Plasma contains different inorganic ions. So these are ions that do not contain carbon, proteins, nutrients, waste, gas, and hormones. In terms of plasma, this is the liquid substance. And so in blood, you have the plasma, and then you have cells. The cells are the white blood cells, the red blood cells, and platelets. So for red blood cells, these are the erythrocytes. They transport oxygen bound to hemoglobin. White blood cells are the leukocytes, and they fight infections, their immunity and defense. Some of those white blood cells are called phagocytes that engulf and digest bacteria or other foreign particles, foreign viruses. Platelets are cell fragments that are involved in the process of blood clotting. So all of these things, you've probably heard of before. You'll want to know these components of blood. And you don't have to memorize the diagram that you see here of plasma and cellular elements, but if this helps you identify and organize the different components of blood, that's why they're here in terms of plasma, what it consists of, and the cellular elements based on cell type. Alrighty. So when there are too few number of red blood cells, you can have a result of anemia that has low amounts of hemoglobin or low number of red blood cells. And since red blood cells have iron in them, with every hemoglobin there's 4 molecules of iron. A measurement of iron amounts in blood can detect anemia. Again, this is not something you need to know in terms of testing. And so here, our checkpoint question asks us, why might increasing the number of red blood cells result in greater endurance and speed? The additional red blood cells have more oxygen, and so that oxygen is supplying to working muscles are able to work quicker and faster. And so there have been some, processes that have occurred for athletes where they essentially are doping on oxygen, and so different processes like suction cups that provide more blood to that area and more blood in general can be used, but I believe they are not approved for official use by any legal sport or official sport. And so athletes that have been doping have usually been in news for being disqualified for that reason. In terms of blood clots are the next thing we'll take a look at, and how they will form structures that stop leakage of blood when vessels are injured. So blood contains self-sealing materials that are activated when blood vessels are injured. And so taking a look at that process on the next slide here. First, there has to be an injury, so the vessel has to rupture in some way. From there, a platelet usually binds to the exposed connective tissue of the vessel. Then from there, this will attract more platelets. So more platelets come and bind to that region. and this forms what we call a platelet plug. And the presence of all the platelets will actually stimulate the fibrinogen to be activated to fibrin, and this will form a fibrin clot that will plug the rupture closed completely until the tissue can be repaired. After the tissue is repaired, then the clot disintegrates. And so in this last section here, it connects the idea of stem cells. So blood is made in the bone marrow. Bone marrow contains stem cells that divide and make different blood cells, like red blood cells, different white blood cells. And so in the leukocytes and white blood cells, leukemia is actually a cancer of these cells where they have uncontrolled growth. There are many other aspects about stem cells, the white blood cells, and even the red blood cells that we haven't covered. But overall, ideas here are explored. And so this slide and then this slide here, along with this one here, is not something I expect you to memorize or to try to understand the complexity of in this section. And so now we've reached the end of this chapter, and which is actually our very last chapter. So this is the last video for lecture. And so I hope you've enjoyed it. I hope you will contact me if you have any questions. And I will be connecting with you online. And so this will be it for now.